Welcome to Inside Drug Discovery. This video describes the discovery program of DD205-291, a PROTAC-based degrader of HPK1 for certain oncology conditions. The discovery program is summarized based on a Drug Annotation article in the Journal of Medicinal Chemistry. The article was authored by scientists at Huadong Medicine in Hangzhou, China. A link to the article can be found in the video description. The target for this discovery program is HPK1, that is hematopoietic progenitor kinase 1. HPK1 plays a role in suppressing receptor signaling in T and B cells. HPK1 therefore suppresses immune response pathways, which can allow proliferation of cancer cells. So the idea is to somehow inhibit the action of HPK1 and better enable a patient to fight cancer through their own immune system. By the way, HPK1 gene knockout mouse models support the described action of HPK1. At this stage, the bigger question is determining the most promising method for inhibiting HPK1. The selected method described in the Drug Annotation article is through a PROTAC approach. PROTAC stands for proteolysis targeting chimera. PROTACs activate a cell's ubiquitin proteasome system, which is basically a cell's garbage collection pathway. A PROTAC drug helps identify a target, HPK1 in this case, as a protein that should be degraded. Structurally, PROTAC drugs have three main components. The first binds the target, HPK1. The second binds to E3 ligase, which is a key player for ubiquitinylation. The third is a tether or linker that connects the other two components. Fortunately for the Huadong scientists, both they and others have published preliminary research in this area. On this slide are two compounds. The left structure, DD203, is an HPK1 inhibitor discovered at Huadong. The right structure, B1, is a PROTAC-based HPK1 degrader discovered by a different group of scientists. Note that the potency of DD203, an inhibitor, is reported as an IC50 value. The PROTAC degrader has a potency reported as DC50. The concentration of B1 required to achieve 50% degradation. The Huadong scientists wanted to use the available information to design a novel HPK1 degrader that shows both selectivity for HPK1 among other related kinases and has adequate oral bioavailability. As you can see, PROTAC compounds like B1 tend to be very large compared to traditional small molecule drugs, and the higher molecular weight can restrict oral absorption. Indeed, despite the amount of excitement around PROTACs as a drug modality, at the time of this video, no PROTAC-based drugs have been approved, although at least one has advanced into phase 3 trials. The discovery program started with DD203 and the exploration of different linkers for the PROTAC-based analogs. Here are three compounds that explore different linkers. The program prepared many more structures than just these three. On the left is the bulk of the structure for DD203 for binding HPK1. On the right is the E3 ligase binder. In the literature, a handful of scaffolds have emerged as suitable E3 ligase ligands, and the Huadong scientists selected from among these more promising ligands. The Huadong scientists did find that this chlorine was critical for potency in the degradation assay. Like the E3 ligase binding region, earlier work on linkers helped guide the options explored. Of the three on this slide, the linker for compound A2 was selected as the most promising, with both excellent potency and extent of degradation. At this point, attention was turned to optimizing the HPK binding region, namely the group on the pyrazole nitrogen. Again, the scientists looked at a number of R groups, with just a few shown here. Most groups were relatively small: methyl, trifluoroethyl, and monofluoroethyl all preserved single-digit nanomolar potency for degradation. These three compounds, as well as A2 from the previous slide, were put through a mouse PK study. Here are the PK data, along with some of the in vitro potency and degradation data. A2 had the best potency and longest half-life, but poor bioavailability. B2 had a very short half-life. B9 and B10 were very comparable, but the maximum percentage HPK1 degradation was higher with B9. With the most promising combination of in vitro activity and preclinical PK properties, B9 emerged as the top lead compound with a research designation of DD205-291. Beyond potency and maximum degradation, the MedChem team had other promising data. First, degradation by DD205-291 was inhibited by known degradation inhibitors that bind the E3 ligase site. DD205-291 also inhibits SLP76 phosphorylation, which is a downstream effect of HPK1. The compound also enhances the release of interferon gamma from T cells, indicating less T cell receptor signaling inhibition. Finally, DD205-291 showed selective degradation of HPK1 over other members of the MAC4A kinase family. While DD205-291 showed some in vivo efficacy in a mouse tumor model, the most promising data were from a 28-day study in rats in combination with an anti-PD1 antibody. In the first row is the vehicle group. The next three rows are increasing doses of oral DD205-291 with moderate benefit of reduction of tumor volume and mass. The anti-PD1 antibody also showed modest benefit as a monotherapy. When used in combination, DD205-291 and the anti-PD1 antibody showed much greater efficacy in terms of reducing tumor volume and mass, with efficacy perhaps plateauing around the 0.5 mg per kg level of DD205-291. With promising in vivo efficacy as a combo therapy and good observed preclinical safety and tolerability, Huadong sought to initiate clinical trials. At the time of publication of the Drug Annotation article, the IND application for DD205-291 had been approved in China and was still pending in the United States. That is the story of the discovery program of DD205-291, a PROTAC-based inhibitor of HPK1. When used in combination with an anti-PD1 antibody, the compound shows in vivo efficacy against tumors in rats with reasonable oral availability. Thank you for watching this video. If you have any comments or questions, please leave them below the video. Also consider liking the video or subscribing to the channel. Take care.